Alrighty, hello everybody. In our last video, we talked about receptor tyrosine kinases. Known as RTKs. And we saw how upon the ligand binding to the receptor in its extracellular domain, that will trigger dimerization of the receptor, and then that will trigger the autophosphorylation of tyrosine residues within its cytosolic domain. Well, for some receptor tyrosine kinase, they can directly then interact with their target protein and phosphorylate that particular target protein, but most don't. And most actually involved a series of basically a signaling transduction that takes place. So you have an interconnected signals that will eventually lead to a cascade, and we'll take a look at the kinase cascade taking place upon the activation of a G protein known as Ras. So we'll also encounter G proteins, right? And we'll specifically encounter Ras in this video, but G proteins are bound to GDP, okay, and that's in their inactive form, and they're activated when GDP is exchanged with GTP, right? Then they're active, right? So we're going to take a look at that as well. So we'll specifically take a look at the Ras signaling cascade, where upon the receptor tyrosine kinase will indirectly activate Ras, and Ras is a G protein. Once it's activated, it's going to replace its bound GDP by GTP, and then that will cause the entire, once it's active, it will cause the entire kinase cascade. We'll notice that Ras is a protein that's anchored to the membrane through prenylation, right? Once it's activated, then it will activate a kinase cascade that eventually will relay the signal into the nucleus. Okay, so let's take a look at what takes place. So if we have a protein growth factor that binds to this receptor tyrosine kinase on the extracellular side, right? This will cause dimerization of the receptor, which in turn will autophosphorylate its tyrosine residues, right? Specific tyrosine residues within the cytosolic domain. Once those tyrosine residues are phosphorylated, the phosphorylated tyrosine residue will bind to a protein known as GRB2. So I'm just going to, the phosphorylated tyrosine residues will interact with a protein known as GRB2. Now, GRB2 is a monomeric protein. It has what we call an SH2 domain flanked by two different SH3 domains. Well, the phosphorylated tyrosine residue will specifically interact with its SH2 domain, right? And it has, it specifically binds to phosphorylated tyrosine and not serine and threonine residue, and that has to do with the actual SH2 domain and the presence of an arginine residue that helps stabilize the charge, specifically of the tyrosine, because of the difference in the distance between phosphorylated serine versus threonine versus tyrosine, right? So they specifically interact with or phosphorylated tyrosine residues in receptor tyrosine kinases. All right, so it'll interact with this SH2 domain, and then it's also flanked by two different SH3 domains that will interact with a protein that is rich in proline residues, and it'll specifically interact with the proline-rich SOS protein. And then we have a complex between GRB2 and SOS that will activate the G protein. Okay, so let's take a look at that. Okay. So now, once the tyrosine residue in the receptor is phosphorylated, and what causes phosphorylation is the binding of the ligand to the receptor, and in this case we're really looking at a protein growth factor binding to its receptor tyrosine kinase, then that will trigger the phosphorylated tyrosine residue to bind to the SH2 domain of SOS, and the SH2 domain of GRB2, and once it's bound to GRB2, what will happen, it will cause the protein SOS to really increase its concentration or migrate towards the inner surface of the plasma membrane, where it will interact with the GRB2. And then this complex will bind to Ras. So Ras is initially inactive and it's bound to GDP. When SOS binds to Ras, it will promote or cause it to exchange its bound GDP by GTP. So GDP and GTP bind to Ras very tightly, and only when SOS is bound is GDP replaced by GTP, right? So really, SOS is activating Ras, right, once it's bound to it. And SOS is bound to GRB2. And now when Ras is active, it will cause this signaling kinase cascade, right? So SOS is known as a guanine nucleotide exchange factor. Or GEF, right? That's the abbreviation. Well, all G proteins typically do have a guanine nucleotide exchange factor that once bound to the G protein, and Ras is an example of a G protein, will help activate the G protein by replacing its bound GDP by GTP. Okay, so now that Ras is active, it will activate, what happens, it binds to another protein known as RAF, and RAF is a serine threonine kinase. It will interact with MEK, activating MEK, right? So RAF binds to the activated RAS only once RAS is activated and is bound to GTP. Once RAF interacts with it, it's a serine threonine kinase, it'll interact with MEK, and it will cause MEK to phosphorylate, right? And once MEK is phosphorylated, it in turn will phosphorylate MAPK at either its threonine or tyrosine residue at a specific sequence, right? Once MAP is also activated, it can then migrate into the cytosol to the nucleus, where it will phosphorylate a number of transcription factors. So that's what we refer to as then the kinase cascade, right? It was all triggered by that ligand or that growth factor binding to the receptor tyrosine kinase. So there was a number of different signals, right? Interconnected signals that led to the kinase cascade that is associated with a number of different proteins. Well, how does that particular signal is controlled, or the magnitude of that signal? Well, Ras itself is actually an enzyme. It can catalyze the hydrolysis of its bound GTP back to GDP, right? Ras is an enzyme, but it can only do so at a rate of few GTPs per minute, right? So there are a number of different enzymes known as GAPs that specifically help accelerate the hydrolysis of bound GTP. So many different G proteins have associated GAPs, and that's GTPase accelerating proteins, right? GAPs. Okay. Right. And that takes care of that particular receptor tyrosine kinase and how the binding of a ligand can trigger this interconnected signals that eventually leads to a kinase cascade, and how we can control the magnitude of that signal one way really by the Ras interacting with GTPase accelerating proteins that will accelerate the rate of hydrolysis of GTP into GDP by 10 to the 5th. All right, I hope you found this helpful, and I will see you next time.